The results of our efforts are molecular machines called base editors. Base editors use the programmable searching mechanism of CRISPR scissors, but instead of cutting the DNA, they directly convert one base to another base without disrupting the rest of the gene. So if you think of naturally occurring CRISPR proteins as molecular scissors, you can think of base editors as pencils, capable of directly rewriting one DNA letter into another by actually rearranging the atoms of one DNA base to instead become a different base. Now, base editors don't exist in nature. In fact, we engineered the first base editor, shown here, from 3 separate proteins that don't even come from the same organism. We started by taking CRISPR scissors and disabling the ability to cut DNA, while retaining its ability to search for and bind a target DNA sequence in a programmed manner. To those disabled CRISPR scissors, shown in blue, we attached a second protein in red, which performs a chemical reaction on the DNA base C, converting it into a base that behaves like T. Third, we had to attach to the first 2 proteins the protein shown in purple, which protects the edited base from being removed by the cell. The net result is an engineered 3-part protein that for the first time allows us to convert Cs into Ts at specified locations in the genome. But even at this point, our work was only half done, because in order to be stable in cells, the 2 strands of a DNA double helix have to form base pairs. And because C only pairs with G and T only pairs with A, simply changing a C to a T on one DNA strand creates a mismatch, a disagreement between the 2 DNA strands that the cell has to resolve by deciding which strand to replace. We realized that we could further engineer this 3-part protein to flag the non-edited strand as the one to be replaced by nicking that strand. This little nick tricks the cell into replacing the non-edited G with an A as it remakes the nicked strand, thereby completing the conversion of what used to be a CG base pair into a stable TA base pair.